Welcome to Deep Dive into Knowledge.
The invisible system that shapes taste.
Welcome to Deep Dive into Knowledge. I want you to pause for a moment and consider the simple act of eating. It is something you likely do 3, perhaps 4 times a day. You do it while checking your emails, while driving, while having a casual conversation about the weather.
Yes.
It is a mundane, rhythmic necessity of staying alive. You place a substance into your mouth, you chew, you swallow. It feels entirely routine.
Quite routine.
But if you were to slow that process down, if you were to truly freeze that moment of contact, you would realize you are engaging in something profoundly high stakes. The instant an object crosses the threshold of your lips, your body initiates a desperate, lightning-fast chemical analysis. You are effectively handing a foreign substance over to a biological security team.
Right.
And you're asking a single binary question: is this sustenance or is this death?
It really is that fundamental.
We tend to believe we are the masters of our own taste. We think, I choose to like this dark chocolate, or I decided I do not enjoy that vintage wine. But looking at the reality of our biology, that feels like a comfortable illusion.
Yes, very much an illusion.
In reality, you are subject to an ancient, invisible machinery that operates entirely without your permission. Before your conscious mind can even register the concept of flavor, a legion of microscopic sensors has already made the decision for you.
Exactly.
They have unlocked chemical gates, fired electrical signals, and cross-referenced millions of years of evolutionary data.
All in milliseconds.
There is a theory suggesting that what you call flavor is essentially a hallucination. It is a construct created by the brain to mask the raw mechanical reality of chemical detection.
A biological narrative.
Precisely. It is a story your nervous system tells you to keep you alive. And if that is true, it forces us to ask a rather lingering, uncomfortable question. If our reality of taste is constructed, how much of the world are we actually perceiving, and how much is simply a narrative written by our biology?
That is the core paradox, isn't it?
It is. Today, we are stripping away the culinary romance and looking at the molecular symphony of taste. I want to begin by looking at the instrument itself. If you stick out your tongue in front of a mirror, you see a rough surface covered in little bumps.
Yes.
We are taught from a very young age that these bumps are our taste buds, but I suspect, looking at the anatomy, the reality is far more complex.
It is indeed, and that is perhaps the most pervasive misconception we have to dismantle right at the start. Those bumps you see on the surface of the tongue are not taste buds.
They aren't.
No, they are anatomical structures called papillae. The taste buds themselves are microscopic. They are buried inside the tissue of these papillae, completely invisible to the naked eye.
So they're hidden away.
Exactly. If you look at the tongue as a landscape, the papillae are the mountains and the forests, but the taste buds are the tiny inhabitants living within caves on those mountains.
So when we burn our tongue on hot soup and shout that we have burned off our taste buds, we aren't actually looking at the damage. We are just looking at the housing.
Precisely. You are looking at the protective architecture. And this architecture is quite varied. We actually have 4 distinct types of papillae, and surprisingly, the most numerous ones have absolutely nothing to do with taste.
Nothing at all. These would be the filiform papillae.
Correct. The filiform papillae. These are the ones that give your tongue that rough, velvety texture. They cover the front 2/3 of the tongue and look like tiny, jagged cones.
Right.
But they contain no taste buds whatsoever. Their primary function is mechanical. It is just friction. They help you manipulate food in your mouth, almost like Velcro.
That makes sense.
If you think of the roughness of a cat's tongue, that is simply an exaggerated version of the filiform papillae we possess.
That is fascinating. The most visible part of the tasting organ doesn't actually taste anything. It is just for grip.
Exactly. The actual tasting machinery is housed in the other 3 types. You have the fungiform papillae, which, as the name suggests, look like tiny mushrooms scattered across the front 2/3 of the tongue.
You can sometimes see those as little red dots, can't you?
You can, yes.
Yeah.
Then, at the very back of the tongue, arranged in a V shape, you have the circumvallate papillae.
Those are the quite large ones at the back.
Yes. They are much larger, dome-shaped structures. They're surrounded by a deep groove, almost like a moat around a castle. And finally, on the sides of the tongue, you have the foliate papillae, which appear as ridges or folds.
And the taste buds are hidden inside these specific structures, inside the moats and the ridges.
Yes, a single taste bud is a cluster of cells. Think of it like a microscopic barrel or a garlic bulb embedded in the epithelium.
A garlic bulb, right.
We have between 2,000 and 10,000 of these structures. At the top of each bud is a tiny opening called a taste pore. This is the gateway.
So the food has to get into that pore.
Correct. Inside the pore, there are hair-like projections called microvilli, or gustatory hairs, which extend out into the saliva. That is where the contact happens.
So the saliva acts as a medium.
It is essential. The chemicals in your food dissolve in your saliva, wash over these microvilli, and that is the exact moment of detection.
This brings us to another image that I think everyone has etched into their memory from primary school biology, the diagram of the tongue.
Ah, yes.
We all colored it in. The tip was for sweet, the back was for bitter, and the sides were for salty and sour. It is such a clean, logical map. But given what you have just said about the distribution of papillae fungiform at the front, circumvallate at the back, does this map actually hold up?
It does not hold up at all. In fact, it is complete fiction. The tongue map is a lie.
A complete lie.
Completely. It was debunked scientifically in 1974 by a researcher named Virginia Collings, yet here we are decades later, and it is still in textbooks. It is still used by wine glass marketers to sell specific shapes of glass to direct the wine to the so-called correct part of your tongue.
If it was debunked in the 70s, how did it become so established in the first place? Science is usually quite rigorous about these things.
It is a classic case of information being lost in translation. It stems from a 1901 German study by a scientist named David Hannig.
Right. Hannig.
He published a thesis showing that different parts of the tongue had slightly different sensitivity thresholds. For example, the tip might be marginally more sensitive to sugar than the back. But, and this is key, he never claimed that the tip only tasted sweet. He simply noted minute differences in sensitivity.
So he was describing nuance, not exclusionary zones.
Exactly. But in 1942, a Harvard psychologist named Edwin Boring took Hannig's data and replotted it into a graph. But he drew the graph in a highly confusing way with no meaningful scale.
Oh, I see.
It made these tiny differences in sensitivity look like absolute zones of exclusivity. Textbook writers saw the graph, misunderstood it, and a tongue map was born.
That is extraordinary.
The reality is that every part of the tongue that contains taste buds can taste every single taste. You can taste bitter at the tip, and you can taste sweet at the back.
It is unsettling how a single bad graph can distort our understanding of our own bodies for a century. But there was a study regarding anesthesia that really drove a nail into the coffin of this map theory, wasn't there? It sounded almost paradoxical.
Yes, this is a finding from 1993 that really illuminates how the system works. The front two-thirds of the tongue is connected to the brain via a nerve called the chorda tympani.
The chorda tympani.
Researchers found that if you anesthetize that nerve, essentially numbing the supposed sweet zone of the map, people didn't lose the ability to taste sweet. In fact, for many, the sensation of sweetness from the rest of the tongue actually became more intense.
That seems entirely counterintuitive. You numb the sensor and the signal gets louder.
It suggests that the taste system is highly interconnected. It seems the nerves inhibit each other to keep the signal balanced. It is a phenomenon called central inhibition.
Central inhibition.
Yes. When you knock out one line of communication, the others compensate or are released from that inhibition. It proves that the system is a unified whole, not a segmented map of independent zones. The brain is constantly synthesizing input from the entire oral cavity to create a stable perception.
I want to go deeper into that unified whole. We have looked at the geography, the papillae, and the debunked map. Now I want to look at the machinery itself. You mentioned that inside these taste pores are clusters of cells. I imagine these aren't just generic cells. What exactly is happening inside a taste bud?
It is a absolute hive of activity. Inside that barrel-shaped cluster, you have about 50 to 100 specialized cells, and they are not all doing the same job. We categorize them into three main types.
Three types.
First, you have the type I cells. These are the most numerous, making up about half the population. They are essentially the support staff for the housekeepers.
Housekeepers. What sort of mess are they cleaning up?
The mess of communication. They provide structural support, wrapping around the other cells, but they also regulate the chemical environment. After a taste signal is sent, there are leftover neurotransmitters floating around. The type I cells degrade or absorb those leftovers to reset the system. If they didn't do this, the nerves would be permanently excited and you wouldn't be able to taste the next bite.
You would just be tasting the ghost of the previous bite permanently.
Precisely. So they are the maintenance crew.
Who are the actual sensors then?
That would be the type 2 and type 3 cells. Type 2 cells are what I like to call the rock stars of the taste bud. These are the cells that contain the highly sophisticated machinery to detect the complex tastes. That is, sweet, bitter, and umami.
Sweet, bitter, and umami.
Yes. They are equipped with G-protein-coupled receptors, or GPCRs.
G-protein-coupled receptors. We hear about those in pharmacology, but how do they function here in the context of taste?
It is a very complex signaling pathway. When a sugar molecule, for instance, floats by and hits a type 2 cell, it binds to a receptor on the surface. This isn't a simple open-door mechanism. It triggers a Rube Goldberg machine inside the cell.
A chain reaction.
Exactly. The receptor activates a protein called gusdeucin.
Gusdeucin. That sounds very specific to taste.
Yes. Gusdeucin then splits apart and activates an enzyme called PLC beta 2. This enzyme breaks down lipids, which releases chemical messengers, which trigger a release of calcium from inside the cell.
It sounds incredibly convoluted just to say, here is some sugar. Why the complexity?
It is convoluted, but it allows for amplification and regulation. A single molecule binding to the outside can trigger the release of thousands of calcium ions inside.
Ah, I see. Amplification.
Yes. Eventually, this cascade causes the cell to release a massive payload of ATP, the energy molecule, which acts as a signal to the nerve fibers.
Right.
And all of this complex chemistry happens in a matter of milliseconds.
And that is just for sweet, bitter, and umami. You mentioned type 3 cells as well?
Type 3 cells handle the simpler tastes, sour and salty. Their mechanism is completely different. They don't use these complex protein cascades. They use ion channels.
Ion channels?
It is much more direct. For salt, which is essentially sodium, the sodium ions just flow directly into the cell through a specialized channel. It is like walking through an open door rather than unlocking a safe.
That sounds much simpler.
It is. The influx of positive charge excites the cell, and it releases serotonin to signal the nerve. Sour works similarly, detecting the hydrogen ions and acidity, which block potassium channels and trigger the same release.
It is fascinating that evolution has designed two completely different engineering solutions in the exact same organ. One is a complex lock-and-key cascade for sugar and bitter, and the other is a simple open door for salt and acid.
It reflects the nature of what is being detected. Salt and acid are simple ionic states, protons and sodium. Sugars and bitter compounds are complex organic molecules that require a specific shape to be recognized. The machinery matches the stimulus.
Let us unpack those stimuli. We traditionally talk about the 5 tastes, but given how complex the biology is, why are there only 5? And why these specific 5? It seems to come down to evolutionary survival signals.
Absolutely. Taste is not about gourmet appreciation. It is about data processing for survival. Each of the 5 tastes answers a specific question about the environment.
What are the questions?
Sweetness is the signal for carbohydrates. Energy. In a prehistoric world where calories were scarce, detecting high-energy food was a superpower. The receptor for this is a heterodimer, meaning 2 proteins working together called T1R2 and T1R3. It is tuned specifically to say, here is fuel.
And salt.
Salt signals electrolytes.
Yeah.
Sodium is critical for nerve function, muscle contraction, and fluid balance. We cannot store salt very well, so we need a constant intake.
That is why a bag of crisps is so addictive then.
Exactly. Your ancient biology is screaming, we need this for our nerves to work. It prioritizes it.
Then we have the warning signals, sour and bitter.
Yes. Sour signals acidity. In nature, acidity usually means one of 2 things. It is either fruit that isn't ripe yet, meaning it has no nutritional value, or food that has spoiled and is teeming with bacteria. It is a caution signal.
And bitter.
Bitter is the master warning system. Bitter signals toxicity.
I have read that our sensitivity to bitter is vastly more complex than the others. We don't just have one way to taste it.
It has to be more complex. There is only one type of sugar, really, that we need to detect. But nature is full of thousands of different poisons. So while we have one main receptor complex for sweet, we have a whole family of about 25 different bitter receptors.
25.
Yes, known as the T2R family. We need to be able to identify a vast array of chemical structures, alkaloids, cyanides, strychnine that could kill us.
That explains a lot about children.
Indeed. That is why we have such a natural, visceral aversion to bitter foods as children. It is a safety mechanism. The brain is saying, do not eat that leaf, it might be poison.
Which brings us to the 5th taste, the one that was ignored by the West for nearly a century. Umami.
The 5th element, if you will. Umami is the taste of glutamate, an amino acid. It signals the presence of protein.
And the history here is quite fascinating.
Very. It was identified in 1908 by a Japanese chemist named Kikune Ikeda. He was eating a bowl of dashi, a seaweed broth, and realized the taste wasn't sweet, sour, salty, or bitter. It was something else entirely.
He knew there was a missing piece.
Yes, he isolated the compound, glutamate, and named the taste umami, which translates roughly to deliciousness or savory.
And yet, I remember being in school decades later and only being taught 4 tastes. Why did it take Western science until the year 2000 to acknowledge this?
It is a mix of cultural bias and scientific stubbornness. Western cuisine focuses heavily on the contrast of sweet and salt. Japanese cuisine is built heavily on umami.
So they just ignored it.
Western scientists dismissed it as just a texture or a mouthfeel. They thought it was a flavor enhancer, not a taste in its own right. It wasn't until researchers at the University of Miami finally located the specific receptor T1R1 plus T1R3 that the debate was finally settled.
They had to find the physical receptor to believe it.
We had to find the lock to believe the key existed.
We cannot talk about umami without touching on the controversy that surrounded it for years. MSG, monosodium glutamate. For a long time, this was treated as a dietary villain, the so-called Chinese restaurant syndrome.
That is a prime example of how a myth can completely overtake science. The term Chinese restaurant syndrome came from a single letter to a medical journal in 1968.
A letter, not a study.
Not a study. It was merely an anecdote suggesting MSG caused headaches and flushing. But subsequent double-blind studies where people didn't know if they were eating MSG or a placebo failed to replicate the results. The symptoms just didn't appear consistently.
So it was a placebo effect. Or perhaps xenophobia.
It is hard to rule out xenophobia. There is a naturalistic fallacy at play, too, the idea that a chemical powder must be bad, while natural food is good. But the irony is astounding.
Why is that?
Because Parmesan cheese contains more naturally occurring glutamate than almost any Asian takeout dish. It's about 1.2% glutamate by weight, yet no one complains of Italian restaurant syndrome.
That really puts it into perspective.
The scientific consensus from the World Health Organization and the FDA is that MSG is entirely safe. It is simply the crystallized form of the umami signal.
We have discussed the input, the chemical hits the receptor, the lock turns, but a chemical reaction on a tongue is not a flavor. Flavor is an experience, it is an image in the mind. How does that raw data get from the tongue to the conscious brain?
This is the journey of construction. The signal leaves the taste buds via the cranial nerves, specifically the chorda tympani for the front, the glossopharyngeal for the back, and the vagus nerve for the throat.
So 3 separate data cables, essentially.
Yes. These nerves travel to the brainstem to a region called the nucleus of the solitary tract. This is the first relay station. From there, it goes to the thalamus, and finally up to the gustatory cortex in the insula.
But the insula just registers sweet or salty. That isn't the experience of eating a strawberry.
Correct, and this is the most crucial distinction in this entire deep dive, the difference between taste and flavor.
Taste versus flavor.
Taste is just those 5 biological signals. Flavor is a multisensory hallucination. What you perceive as the flavor of a strawberry is actually about 80% smell.
80%? That seems incredibly high. We think of eating as an oral experience?
Roughly 80%, yes. But it is a specific type of smell. When you chew, volatile molecules float up the back of your throat into your nasal cavity. This is called retronasal olfaction.
Retronasal, so smelling from the inside.
Exactly. Your brain combines that olfactory data with the 5 taste signals, plus the tactile sensation of texture, plus the temperature, and even the pain reception.
Pain reception?
Yes, like the burn of a chili pepper. That is capsaicin triggering pain receptors, not taste buds.
So the brain stitches all these disparate inputs together into a single seamless experience.
It does. This integration happens in the orbitofrontal cortex. This is where the sensory input meets emotion and memory.
So that is why context matters.
Very much so. It is why food tastes better when you are hungry. The reward circuits are active. It is why a meal tastes better when you are happy, or why a certain soup might remind you of your grandmother and actually taste like comfort. The biology provides the raw data, but the brain writes the story.
This construction of reality implies that my reality might be different from yours. We assume we all taste the same apple, but the biology suggests otherwise.
We absolutely do not taste the same apple. We are all living in slightly different sensory worlds.
Can you give an example of that?
Well, there's a phenomenon known as supertasting.
Ah.
About 25% of the population are supertasters. Anatomically, they are built differently.
How so?
If you put a small circle on their tongue, you might find 60 or more fungiform papillae, whereas a non-taster might have fewer than 30 in that same space.
That sounds like a superpower, but is it actually pleasant?
Often, it is not. Because they have so many receptors, the signal is incredibly loud. Bitter tastes, in particular, can be overwhelming. Supertasters often find coffee unreadable, alcohol repulsive, and vegetables like broccoli or Brussels sprouts chemically abrasive. Their world is turned up to maximum volume.
And this is purely genetic.
Largely, yes. It is linked to the TS2R38 gene, but there are other genetic quirks as well. Take cilantro, or coriander as we often call it.
Oh, the great coriander divide.
Yeah, to most people it tastes fresh and citrusy.
Uh-huh.
But to about 10 to 14% of the population, it tastes exactly like soap.
I have friends who insist on this. They say it ruins the entire meal if there is even a leaf of it.
And they aren't being difficult or picky. They have a genetic variation in their olfactory receptors, specifically the ones that detect aldehydes.
Aldehydes.
Yes, coriander is full of aldehydes, but so is soap. These people are genetically hardwired to pick up that specific soapy chemical note that the rest of us simply miss.
It is genuinely humbling to realize that our preferences, which we think of as personality traits or sophisticated palates, are often just the result of a genetic dice roll. But surely we aren't slaves to our genes. I hated coffee when I was young, and now I rely on it.
That is the plasticity of the system. We can override the biological warning. Coffee is bitter. Your body's T2R receptors scream, poison.
But we drink it anyway.
We do, because your brain learns that the caffeine payoff is positive. Through repeated exposure, you condition the brain to associate that bitter profile with a reward. You are effectively hacking your own survival software.
I want to look at the timeline of this software. You mentioned earlier that these cells are working hard in a harsh environment. They're dealing with acid, heat, physical friction. They can't last forever under those conditions.
They don't. The lifespan of a taste bud is incredibly short, only about 10 to 14 days.
2 weeks.
Yes, you essentially get a brand new tongue every 2 weeks. Stem cells at the base of the bud constantly divide and mature to replace the dying cells.
Does this renewal process keep working perfectly as we age?
Unfortunately, no. It slows down. We start life with that full complement of up to 10,000 taste buds. But as we age, the regeneration simply doesn't keep up with the cell death.
How significant is the drop?
Very. By the time someone reaches age 70, they may have lost 60% of their taste buds. They might be operating with only 4,000 active units.
That explains so much about the changing dietary habits of the elderly.
It does. You often see older people adding excessive salt or sugar to their food. It is not that they have suddenly developed a sweet tooth. It is that the signal is fading. They need a louder shout to hear the whisper.
It must impact their quality of life.
Profoundly. It explains why appetites fade in old age. Eating becomes less rewarding because the sensory symphony has become a very quiet solo.
And it is not just aging. We have seen recently how fragile this sense can be with illness. COVID-19 brought the loss of taste and smell into the global spotlight.
It did. And for many, it wasn't just a loss, which is called ageusia. It was parosmia, a complete distortion.
Parosmia. I remember reading about people who couldn't stand their favorite foods.
Yes, familiar smells and tastes became twisted. Coffee smelling like burning rubber, or meat tasting like rot. It showed us how delicate the neural connection is. If the virus damages the olfactory neurons, or the support cells in the tongue, the brain starts receiving garbled data.
It tries to make sense of the noise.
It tries to interpret it, but the result is a nightmare. It is a similar tragedy with chemotherapy. The drugs target rapidly dividing cells, which unfortunately includes the stem cells replacing taste buds. The buds die, the food tastes metallic or like cardboard, and the patient loses their appetite right when they need nutrition the most.
It really makes you realize how much we take the mundane pleasure of eating for granted until the machinery breaks. But there is one final twist in this story that I found the most surprising of all. We have been talking entirely about the tongue, the mouth, the nose. But looking at the research, it seems these receptors, these T1Rs and T2Rs, are not actually limited to the mouth.
This is truly the frontier of taste science. We have found taste receptors all over the body. We found sweet and umami receptors in the lining of the intestines.
In the gut. What are they tasting down there? It is entirely dark.
They are tasting the nutrient content of your meal. When the gut receptors detect sugar or glutamate, they signal the release of hormones like insulin to prepare the body to absorb the energy. They are regulating your metabolism before the food is even fully digested.
So the gut is tasting the food to decide how to process it. That is incredible.
Exactly. And it gets stranger. We have found bitter receptors in the lungs and airways.
In the lungs.
Yes. Now, they aren't connected to the conscious part of the brain. You don't consciously taste the air.
Thank goodness for that.
Indeed. But if you inhale bacterial toxins, which are often bitter, these receptors trigger a local defense response. The little hairs in your lungs start beating faster to clear the mucus. It is an immune response triggered by taste receptors.
That is extraordinary. The body is using the exact same lock and key code in completely different organs for completely different purposes.
It goes even further. Bitter receptors have been found in sperm cells.
I beg your pardon?
It is true. We aren't entirely sure of the specific function yet, but it may be involved in chemotaxis.
Chemotaxis, navigating by chemicals.
Yes, navigating the sperm toward the egg. It seems that taste is not just a culinary sense at all. It is a fundamental, body-wide chemosensory system. We are tasting our environment from the inside out, constantly.
It fundamentally shifts how we view the body. We are not just a mind riding around in a vehicle. The vehicle itself is intelligent. It is constantly sampling, testing, and reacting to the chemistry of the world.
We are merely the conscious passengers. While we sit here discussing the flavor of a fine wine or a complex dish, our intestines, our lungs, even our reproductive cells are having their own silent conversation with the chemical reality around us. We are immersed in a chemical ocean, and every part of us is designed to read the currents without us ever knowing.
That is a profound place to land. We started with the idea that the tongue map was a lie, and we have ended up realizing that the tongue itself is just the tip of the iceberg. The bumps aren't buds, the flavor is a hallucination of the brain, and our lungs are tasting the air for poison.
It certainly changes your perspective on the next meal.
It creates a new level of respect for the act of eating. The next time you sit down for a meal, I invite you to pay attention, not just to whether it tastes good, but to the mechanics of it. Try to separate the smell from the taste. Ask yourself if you are sensing the texture or the flavor. Try to feel the machinery working. Thank you for joining us on this deep dive into the molecular symphony of taste. We will be back soon with another exploration into the hidden layers of our reality. Until then, stay curious.